Everyone is familiar with the fact that when you look at a flora nowadays, a modern flora, there are all sorts of strange names of plants, and there are all sorts of strange reorganizations of plant families. And this is, of course, disconcerting because we've actually had a period of relative stability in our nomenclature due to the fact that the people that wrote the major floras like Gray's Manual and the Gleason and Cronquist Manual just haven't been superseded. So we've been very conservative here, but I think that has changed. New floras that are coming out now are dramatically different. And I thought that I would put this together to illustrate some of the reasons why some of our names are changing and some of the theoretical underpinnings that underlie those changes and why it's important that we make them and why once you get used to it, you'll feel a lot better about it and they'll actually enrich your experience with the plants. So the talk is really in two parts. Very briefly, I'm going to talk about the naming of plants and what plant names mean. And then the second part, which is also responsible for changes in names, is discovering how plants are related to each other. How do we do this? So, okay, first of all, the naming of plants. You are all familiar with the statement that a plant can have only one correct scientific name. The scientific name is composed of a genus and species. The thing that's often forgotten in this is what's tacked on at the end when classified in a particular way. So what that basically means is if your classification changes, the name may also change. So keep that in mind. When people tell you that scientific names are stable because every plant can have only one correct one, that's true for any particular classification. Of course, Linnaeus started all this with Species Plantarum in 1753, and his great contribution was the condensing of complex polynomial names by which plants used to be known in the Mediterranean era into these binomials. And so that was the great benefit. And the other great thing that he did was arrange a hierarchical classification. What does that mean? It means that plant names are in ever decreasing units, each unit subsuming the smaller ones. So there's a plant kingdom, a division, flowering plants, class, Rhosopsida, the true dicotyledons, the buttercup order, the buttercup family, the genus, and the species. And the only name that we really use popularly are genus and species, and of course we classify things into families and key out families. You never see orders in books to any extent. So what this means is the plant name is linked to this hierarchy and encapsulates it, and any particular species will have the features of all of the upper level groups. So what this means naturally is that if the classification changes, if you decide that a plant goes in a different hierarchy, then the name may change, at least if the different hierarchy involves the genus and species. So, and of course, it may also go into a different family, which is also disconcerting. So that's basically the underpinnings of why names can change despite this very stable organization that we have built upon since Linnaeus. And one thing that I like to point out is all of our modern nomenclatural and taxonomic things we see build on the genetic relationships of plants and how they are intertwined with each other in their relationships. But this classification actually occurred before Darwin, before genetics was understood, and before we understood how inherited variation drives evolution. So the science was based on correlating discontinuities and patterns of variation. And that works surprisingly well. It's actually, to me, in many ways amazing how well it worked. But you can easily be fooled by superficial similarities or when plants differ radically in a few very striking features. And I'll show you some examples of that later on. So now we think we are getting closer to the answer. So, but why are we sure of this? Why won't we, in 30 years, discover that everything we thought is hogwash? Well, I think I'm optimistic that that won't happen. And the reason is actually a couple of reasons. One is, of course, modern systematics has a much deeper and more comprehensive understanding of variation. We know about genetics, we know about inherited characteristics and characteristics that are not inherited and just environmental. But actually, the most important thing for understanding plant relationships are three particular items. First off, we can access the genome directly. And you are basically a rather liquid, sloppy bag that genes use to regenerate themselves. So we think that by being able to access the genome directly, rather than worrying about all the variation we see in those fleshy bags that contain the genome, we're getting much closer to the core of what makes you a member of Homo sapiens, or what makes a Carex species a particular entity. Now, of course, the genome is big, and there's a lot of features that it has. So in addition to accessing the genome, you need to figure out what to do with it. And there are two other things that relate to that. First of all is mathematicians have developed complex techniques that allow them to take differences in the genome and construct a probable branching pattern that produced those differences. And not only that, to give them even some statistical significance. Now, few taxonomists actually understand these complex algorithms. They're generated by mathematicians that sit in closed rooms and smoke too much. I don't really know. In any event, they're rather useful. But until rather recently, they were also somewhat pointless, because the calculations as applied to our genetic material, which is so massively complex, couldn't be done. But now we've actually developed powerful computers that can perform these mathematical analyses. So we can actually derive branching patterns that give us an insight into relationships, ancestor-dependent descendant relationships in the past. So there's DNA. There's the base pairs that make up this chain. It's of course twisted into a spiral. And there are millions and millions of base pairs in your genome. More than even Donald Trump can imagine. It's huge. So, isn't that awful? My God. Imagine reminding people of the Republican primary. I'm sorry. In any event, if you think about what you do when you identify a plant, you use some characteristics and you measure the leaves and the height and a bunch of stuff. How many characters do you think your basic plant has? A few dozen if you're really thorough and do anatomy and cellular measurements and that, you might actually get up to a couple hundred. But that plant's genome will have millions and millions and millions and millions of characters, base pairs. They're basically a character like measuring the width of a leaf or looking at the color of a flower. And it's those millions, those huge numbers, that give us confidence that we're getting closer to the right answer. So these equations that allow the reconstruction of these evolutionary branching patterns produce a diagram kind of like this. And you have to think of these diagrams as hypotheses, but they're pretty solid, and this is the kind of thing that you can look at. People carelessly say things like, we're descended from apes, and apes are descended from monkeys, and monkeys are descended. By the time you get down there, you're descended from crayfish and all sorts of things. But in actual fact, what really is the case is that mammals and all these things share a common ancestor long in the past. We're not really descended from crocodiles or anything ridiculous like that. They share a common ancestor. So, and there he is. See, until computers became a little less primitive than that, we couldn't do these calculations. So this is kind of what we're getting at. And of course, this is a very simple diagram. And when you take a diagram like this and interpret it to make a classification, you're actually just looking at the tips of the diagram. So how do we interpret these phylogenies? Well, here we go. This is the pattern we get with an evolutionary tree, a very simple one. So how do we turn this into a group forming thing? Well, it's very simple. There's a group. There's a group. That looks good. We like that. We can name these things, some genus and species. Well, there's a few problems. This would also be correct. So you have still differences of opinion. Can't have science without differences of opinion. So this is how we would interpret these phylogenetic diagrams. And of course, in reality, they look much more complicated, and this is on purpose. Of course, scientists do this because you can't really present a good seminar unless you have some inscrutable slides. So this is the kind of thing that's very important. Now, how does this look when you look at plant families and plant orders? And let's look at some interesting things about that. There are some really cool things that we've learned when we have taken all that DNA evidence and basically run the entire green plant system through it. First of all, keep in mind that way down here is the end point, and the things come off this. The lower down they come off, the longer ago they shared a common ancestor. So way back near the bottom, sticking off this somehow, are ferns. And there's a fern. We all like these little guys. And then, of course, next in line, now this is nothing unusual, next in line, of course, are conifers, along with cycads, ginkgo, and the Netales. This is more high school and undergraduate botany. Nothing too unusual there. But now there's something really interesting that we never knew before that I think people can appreciate. How many of you learned about monocots and dicots in school? You remember that? No, monocots have parallel veined leaves and flower parts in threes, and dicots have netted veined leaves and flower parts in fours and fives, and they have two cotyledons instead of one. Well, it ain't so. We all started out as dicots, and somewhere right there, the monocots branched off, and then there's the rest of the dicots. So dicots actually are composed of two different things. What we traditionally call true dicots here, and then these primitive things down here that predate the split between dicots and monocots. And these are pretty cool plants. I really like these guys. There are a lot of interesting things among these. So what are some of them? Right there are several lines going up. Amborellales, what on earth is that? Nymphaeales, that we know. Water lilies and their cousins. And Austrobalaceae, what on earth is that? This is a group of families that is referred to by this name right here. That's the first letters of the families in there. These are ancient relics, and they actually predate even the split between monocots and dicots. So, and there are a couple of interesting things about these. One of them is, of course, that people generally thought that the primitive plant would be something dinosaurian, like this magnolia. Big, floppy thing. You can imagine some dinosaur snout snarfing down those leaves and flowers, and it all fit. Well, it's not that way. Actually the most primitive plant extant now is this little unisexual tropical shrub. Hard to believe. So it's a real shift in our thinking. So what else is in this basal group way back? Elysiaceae, these two. Water lilies, yes, we know water lilies. Magnoliids are also primitive, and they include not only dinosaurian things like magnolia, but also another interesting and unusual sidelight to our understanding of flowering plants, and that is that not all these extremely primitive things were woody. A lot of them were herbaceous. So the word of the day is paleo herb. a lot of these primitive things were herbaceous plants. And they have some really unusual features that I'll mention. Well, one of them is this. That's a paleo herb. Next time you're walking through the woods in the spring, you can point that out to people, and you'll amaze your friends and confound your enemies, and it'll be great. There's an interesting twist. In retrospect, botanists should be slapped silly for not realizing that there was something interesting about these primitive plants that we used to think were dicots. How many people can guess what it is? What's the common factor in all these primitive flowering plants that we used to think were dicots? What's unusual about all of these flowers?
3 parts.
3 parts. Exactly right. Chloranthaceae. That stripped down, just stamens. Sassafras. How many petals does sassafras have? 1, 2, 3, 4, 5, 6. Pawpaws. Look at that. 1, 2, 3. 1, 2, 3. Brassinia. 1, 2, 3, 4, 5, 6. How many stigmas? 1, 2, 3, 4, 5, 6. Tulip tree. 1, 2, 3, 4, 5, 6. 1, 2, 3 sepals. Nobody ever noticed this, really, and put it together, that these things actually predated the split between monocots and dicots, and a mark for that was that their flower parts were often in 3s. Even the monocots have something interesting about them. Acorus, the sweet flag. This is a very important plant economically. The European sweet, used to be anyway, the European sweet flag, which is actually not our native plant, although it's introduced a little bit in Michigan, was highly used in medieval days because people didn't wash, and so they, instead of using deodorant, they put sweet flags out in all the rooms and things. And when you see phrases in old books, I think even Shakespeare, about laying down flags, well, that's what they meant. They put sweet flags so you couldn't be smelled. So Acorus is not an aroid. It's not a member of the Araceae family, which is what we once thought. But it turns out it's the most primitive of all monocots. So that's a really interesting feature. In the Acoraceae, yeah, there's only one of them. It's an ancient relic surviving from the most primitive times. So, all right, what's all this got to do with name changes? Well, there's more to what we learn with reconstructing phylogenies than just being able to go deep into the past and understand how plant families are related. And in fact, it's also influenced how we divide plants into families, and also how we divide plants into genera, and that's where name changes come in. So what's behind these name changes? One of the things that you can do when you can divide plants into groups and have a reliable ancestor-descendant relationship is you can start thinking about what makes a sensible group and what doesn't. And that is what's behind a lot of the name changes. Back to this diagram. So I showed you how groups were formed from that, but let's look at what happens when you get inappropriate groups, and that's really what underlies a lot of the changes in names. If you have a group that includes these two And then this interloper, that group is actually formed of two separate lineages. If you think of a human family in terms of strictly genetics, this fellow is adopted. Is not going to share the same genetic background or the same ancestors as you do. And a group like that, that's formed in this way, is called polyphyletic. That's a no-no. We don't like that because we like our plant families and our plant genera to be just like human families. No adoptions. You can also get one other way of transgressing good classifications. Let's say somebody decides that this thing right there is striking and outstanding and remarkable. Maybe it's hummingbird pollinated. Maybe it's delved into an aquatic environment and looks very different. So they pull this out as a separate group, and then they put these two together in the same group. We don't like that either, because a group not only should be all descended from the same ancestor, in order for the formation of such a group to not be simply arbitrary, you also have to have the group contain all of the descendants of some particular ancestor. You can't pawn off somebody onto another group, even if they're the black sheep of the family. So a group that's formed that way, including some but not all of the descendants, is called paraphyletic, and we don't like that either. And it's those two concepts that unfortunately were relatively common in classifications that were built prior to our ability to access the genome. So a group that in fact contains all of the descendants of a single ancestor is monophyletic. A single line is what that translates to, and that's what we like. So when we have gone back and corrected the classifications to make them all monophyletic, that is basically accounting for most of the changes. So if any group, like a family or a genus, doesn't all come out together in one spot, when its phylogeny is reconstructed, that's a problem. So here's the kind of thing you end up with. We all like our nice boreal black crowberry, very rare in Michigan, just on Lake Superior. Well, here's a phylogeny of the heath family. Very nice family, everyone likes it. Blueberries, all sorts of cool things in there. But look at what's sitting right in the middle of the blueberries. Empetrum. Now, how can you have a family nestled deep within another family? It just doesn't make sense. And of course, what we would say is that recognizing Empetraceae as a family makes the Ericaceae paraphyletic because it doesn't include all of the descendants of the common ancestor of that family. So we wouldn't recognize Empetraceae. We would just dump it into the Ericaceae. And it looks just like an Ericaceae. Why was it separated? It has a unique pollination system, unlike other Ericaceae. It's wind pollinated. So if you're wind pollinated, you don't need to fuss with any of that fancy stuff for attracting bugs like petals and nectaries and all of that stuff. You can strip yourself down to just your stamens and your pistils, but your flowers look so incredibly different that even though the plants and the fruits are the same, people would put it into a different family. So that's what happened. So now we dump Empetra back into the Ericaceae. Lilies. Same thing happened with lilies. When people looked at the phylogeny of lilies, it turned out they were actually what we used to call Liliaceae. It turned out they were actually in several different orders, much less families. And actually, in retrospect, when you look at them, they don't look remotely alike. So we have all these things now. This is still Liliaceae. Allium is in the Alliaceae or Amaryllidaceae. Elytris is in the Nartheciaceae, and so on. So we've divided the lilies up to make each of the lily families monophyletic units. Lots of families turned out to be pretty heavily impacted. Scrophulariaceae no more. Penstemons are actually plantains with makeup. They're plantains that have made up their flowers to look gaudy and showy to attract bugs. So that's kind of surprising, but the molecules don't lie. So there's your penstemon, and there's your plantain. There's a bigger plantain. You're excused if you don't see the resemblance. It's in their genes. And there's actually a lot more to it than that. These Scrophulariaceae, what used to be Scrophulariaceae, are all hemiparasites. Hemi because they still have some green color, but they all parasitize other plants. And it turns out when you look at their classification, they actually fall together with these plants, which we used to put in the Orobanchaceae and still do. Why? Because these don't have any chlorophyll or leaves or anything. They're total parasites. But actually, when you look at these hemiparasites, they're not all that green to begin with. And now we are very sensibly put them together in the same family. All the parasites go together, whether they have green leaves or not. So, very good. This is the oddest thing. Monkey flowers and our eastern woodland lopseed. You know, who would have ever thought? Their fruits are so dramatically different. On the other hand, if you look at the flowers, that doesn't look that different. And the leaves look kind of the same too. So that's okay. That's a pretty odd alignment. What do we have left in the scrophs? Ooh, verbascum, mullein. Well, of course, Scrophularia too. And Buddleia, the butterfly bush. Where does that come in? I have no idea. But its genes don't lie. So that's the kind of thing that we see when we look at phylogenies. Honeysuckles, Adoxaceae for Sambucus, and Caprifoliaceae, now restricted to honeysuckles and their very close relatives. There is a pattern when you look at these changes. There really is something that you can predict. Highly variable families that were basically junk heaps of all sorts of different looking things thrown together, they turn out to be polyphyletic, unnatural. Families that are easily recognized and have a distinctive, unique adaptation often turn out to be monophyletic. Small families that are highly specialized and reduced, like aquatics or wind pollination or hummingbird pollination, are often widely misplaced in classification systems. People zeroed in on the weird, strange characteristic and used that to establish a family, and it just ain't so. They just diverged in that one feature. And commonly too, unusual temperate representatives of widespread tropical families were often also separated out as their own family, and that's not so either. So here's a great example here. Calitrichae, here's the smallest one. Man, it is so small, it looks like a little tiny seedling. But when you get down in there, there's the flowers. Nobody's going to write home about that. There's the stamens, there's the pistil, and they develop these weird two-lobed fruits. You got to get really close. We used to put this in the Calitrichaceae, but it turns out it nests right into the plantains, and these extreme differences. And remember, plants are very prone to this because plants are very plastic. These extreme differences are the result of adaptation to an aquatic or semi-aquatic environment. This thing grows in, well, it typically grows in tire ruts and wet roads. So that's, millions of years of adaptation to growing in tire ruts is kind of an interesting thing. I don't know how that works. But anyway, it's caused it to have its classification be very difficult to really assess. Of course, unfortunately, lots and lots of genera haven't been examined yet. And sometimes the changes are kind of cool. Like there's our ledum. And if you look at that, it's nested right in among the subgenera of rhododendrons. And sometimes we've understood this even before we looked at the molecules. So ledum has to go. That's a good thing. I mean, Michigan now has a native rhododendron. What's bad about that? Isn't it great? So, and there's what happens when you look at the molecules. All rhododendrons, bang, there's ledum. All rhododendrons, well, there's another genus. So you can see this is nested right in the middle of it. You can't pick that out and leave all the rest as rhododendrons. That doesn't work. But there are lots of good things, or at least relatively small changes. Irises. I mean, once you've seen one iris, you've seen them all, and they all stay together quite nicely. Well, there is a little bit of a difference. This iris, which doesn't look much like an iris until you look close, our blackberry lily is now not Bellum candida chinensis, but Iris domestica. And it's nestled right in, where is it here? Right there, smack in the middle of the irises. So its flowers are a bit funny, and so are the fruits, but it's an iris. There it is. Bottom line, it's your genes that matter, not your phenotype, not your external appearance. So, yeah, where?
That daisy will never be the same.
It'll never be the same, no doubt about it. It's amazing what you can dig up when you hunt around. That's why they train people to do bibliographic research properly. So, which actually generally doesn't include reading Mad Magazine, but in this case, we're good. So, asters. This is a real sore point. Asters. Ooh, the aster disaster. What's the problem with asters? Well, here you have a nice phylogeny, and here are all the places where what we used to call asters come out. You know already that this is bad. That's why we have all sorts of fancy new names for our asters. And of course, the rules are such that any substitute name has to be several syllables longer and much more difficult to pronounce. So there's some of these unwritten rules here. So that actually is one of the most striking changes that we've had to make. But there are lots of families that aren't changed. Mustards. Mustards are all the same. Once you've seen one mustard, you've seen them all, good or bad. Beans. These striking specialized flowers are a cohesive natural unit, and they stay that way. The family that asters belong to. Once you've seen one daisy, you've seen them all. I mean, they're all the same. The Asteraceae stays as a nice natural unit. Orchids. Orchids are all a natural unit, and the families are all uniform and stay that way. So that's good news. Grasses. Grasses all hang together. You know, by the time you add up all the grasses and all the orchids and all the Asteraceae and yes, indeed, all the sedges, which are also a cohesive unit that sticks together, none of these have had their family affinities change. So most of the flora basically has stayed the same. So that's not all bad. That's actually pretty good. So you know, we're in pretty good shape with a lot of things. So, but the asters we definitely have to apologize for. So anyway, why don't I just stop and we'll get some questions for.